Hi, welcome to my channel. Today in medical research, we're going to talk about cancer genomics, the suppression of cancer through microRNAs. In the introduction, I want to talk about cancer therapy, some traditional methods, what is genomics, also talk about targeted therapy and microRNAs. But the main question for microRNAs is how can microRNAs be used for cancer therapy? So some traditional methods that we know is chemotherapy and radiation therapy because they are still used as of to date. Chemotherapy is the use of medicines or drugs to treat a disease such as cancer. Radiation is the use of high energy radiation to shrink tumors and kill cancer cells. Chemotherapy and radiation therapy can either damage DNA or create charged particles, which we know it as free radicals. The free radicals within the cell that can in turn damage the DNA. They can also damage normal cells, which can lead to side effects. Now, I do want to say this. I'm not against chemotherapy. I know a lot of people and have seen proven that a lot of people's life has been prolonged with the treatment of chemotherapy. However, there are a lot of people too that chemotherapy has not worked on, and I do believe this is because they are further along with their cancer, and the chemotherapy isn't enough to eliminate the cancer cells. So what is genomics? All the DNA contained in your cells make up your genome. Let's think of all our DNA as an alphabet. So with that being said, genomics is the study of the sequence of letters in your DNA. So when you get small changes, you get diseases such as cancer. Targeted cancer therapies are drugs or other substances that block the growth and spread of cancer. Now, the good thing about targeted therapy is it can act on a specific molecular target. It is deliberately chosen or designed to interact with their target. So it's not like chemotherapy where it damages your good normal DNA or normal cells. It only affects its target. And they are often cytostatic, which means they block tumor cell proliferation. MicroRNAs, just a quick briefing on what are microRNAs. They are short, single-stranded RNA molecules. They're approximately 22 nucleotides in length. And microRNAs expression, one thing about expression is it varies dramatically across tumor types. Whatever you could have lung cancer, the microRNA that is expressed there is different from breast cancer. So keeping that in mind, we will move on to the results. Here's the first paper. This is the title. At the bottom, you can see the authors.
This experiment was done in vitro. MicroRNA was injected and it is suppressing the growth of colon cancer. The non-control at the top does not have the microRNA, but the mimics at the bottom do contain it. Looking at the top row in comparison to the bottom row, you can see a clear suppression in the growth of the tumor. Also here you can see the word inhibitor in the row to the far right, and this shows the natural growth of the tumor when RNAs were completely inhibited. Now here we have tumor growth in vivo. The mice were injected with LOVO line of the microRNA 520D. The vector is the control. It does not have the microRNA. After a 22-day observation, you can see that the mice with the microRNA injected tumor is smaller than the vector.
Here is the second paper, and we will be looking at microRNA 33A, which is should inhibit the lung cancer.
This shows that transfection with microRNA inhibits the proliferation of cancer. This example is in lung cancer. The white bar is the control that does not have the microRNA, and the black bar contains microRNA 33A. During the 96-hour period, you can see that the growth of the cancer decreases.
In conclusion, microRNAs can suppress cell proliferation, which can result in the inhibition of growth of cancer cells and tumors. So microRNAs may be used for targeted therapy as a replacement for traditional methods such as chemotherapy, or it could be used, I think it could also be used with chemotherapy. For example, those patients that are real far along with their cancer that chemotherapy doesn't work, how about we target it and shrink the cell down and then, eliminate it with the chemotherapy. I think that is something that we could try, and it may be coming in the near future. Everything will be in the description box. If you want to read the papers, I'll link them in the box below. My email, Twitter, all of that will be linked. And if you like this, subscribe, thumbs it up. And until the next video, peace, love, and happy thoughts. Bye.